Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. In this video lecture, we are going to talk about protein synthesis inhibitor. Fine. Our view of the protein synthesis inhibitor. Fine. That what are actually we are going in our next video lecture. Fine. So in the previous video lecture, if you remember, we talked in a detail about the nucleic acid inhibitor. Fine. So this is another video lecture which are regarded with antibiotic drugs, and in this video lecture we are going to talk about protein synthesis inhibitor. So let me write protein synthesis inhibitors. Protein synthesis inhibitor usually in prokaryotic cell. In prokaryotic, or we can say in a bacterial cell. Bacterial cell. Fine. Protein are very important, or we can say protein synthesis process are very important for the bacteria surviving. Fine. Bacteria use this protein for different kind of purpose, fine, inside the bacterial cell. Now how the protein are synthesized and what are the main drugs which are used in a protein synthesis inhibitor, let me explain. If we consider the central dogma of biology, so what actually it tells us about? It tells us about that let's suppose this is a DNA. So what we get actually from DNA, we get the RNA, and from RNA what we get the protein. So these are the protein. This process is called translation. And the drugs will also no, the protein synthesis inhibitor, the drugs which are used in protein synthesis inhibitor, they usually target to that area, or we can say they block to that area by which the protein are not synthesized. Fine. If we see that how actually the protein are synthesized, so there are three main things is involved in a protein synthesis. Fine. The first thing is messenger RNA, the second thing is ribosome, and the third thing is the transfer RNA. Due to these three things, a protein will be synthesized. Now, before going to explain that how a protein is synthesized, first of all, let me write here some important drugs which are used in a protein synthesis. Now, there are some important drugs which are used in a protein synthesis. These drugs are the tetracycline, This is the first drug. The second drugs which are used in a protein synthesis, inside the tetracycline there are some other. This is the name of the group. This is the name of the group and there are some drugs which are used in a tetracycline. Inshallah, we'll be talking in next video lecture. And there are another drugs which are used in a protein synthesis. This group of drugs is called macrolides. And there are also some drugs which are also used in a protein synthesis inhibitor. These are the aminoglycosides. So these are the drugs. Fine. Now, this tetracycline is actually used to inhibit Now, listen, let me tell you one important thing, that for protein synthesis, the ribosome play a vital role. Okay? Why? Because ribosomes contain three sites. Inshallah, I will talk in this video that what actually happened. A ribosome contain a three site, P site, E site, and A site. Due to this ribosome machinery, there will be a transfer RNA are coming, and then the ribosome will move forward, and then the protein usually gradually the protein will synthesized. So we can say that here the ribosome is actually play a vital role. Once we stop the ribosome, so the whole process, or we can say the protein synthesis process, will be stopped. So we can say that these drugs will actually attack or target the ribosome. So each of the drugs is actually targeted. Each of drugs is actually targeted the ribosomes. Now if we see in our cell, or we can say in eukaryotic cell or mammalian cell, there is also ribosomes present. But why these drugs are attacked in a bacterial ribosome and why they are not attacked in a eukaryotic ribosome? The reason behind that, they can attack just on the ribosomes of the bacterial cell because the bacterial cell is made up of 70S ribosomes. The large subunit is 50S and the small subunit is the 30S. In case of eukaryotic cell, it is made up of 80S subunit of the ribosome. The small is 40S and the large is 60S subunit. Fine. So what actually happened? This tetracycline, now this ribosomes is actually 70S subunit. Fine. What actually happened? The 50S and 30S are bind together, then they make 70S ribosome. Now this tetracycline will usually attack on 30S. They attack on 30S to stop the protein synthesis. This macrolides will attack on 50S to stop the protein synthesis, and this aminoglycoside will attack on the 70S ribosome to stop the protein synthesis. So these are the drugs, and these are the mechanism of action of these drugs, and these are the target site of these drugs. Inshallah, we'll be talking in a detail in our next video lecture. Now, before going to explain the mechanism of action of each drugs, first of all, we need to understand that how actually the bacteria synthesize their protein. Once you get the synthesis of the protein, then the videos which are regarded with the mechanism of action of tetracycline, macrolides and aminoglycosides will be very easily for your understanding. So protein synthesis, or we can say translation process, is a very complex and very complicated and a very detailed topic. Fine. It takes almost 2 or 1 hour when I explain it. Now, and it also contains some videos lecture when I explain it. Fine. So inshallah, in this video I will just a little bit touch it, not explain it in a detail. I will just briefly introduce it, that what actually happened, or we can say that I will just give you main idea about the protein synthesis, and then inshallah in some other videos lecture, then we will talk about the protein synthesis in a detail. What actually happened for the protein synthesis? As I told you that we need 3 main things. The first thing is messenger RNA. This is the first thing. We call that messenger RNA. This messenger RNA contain codon, or we can say information thing. This information thing is actually called as codon. So let me draw here with a black marker. So these are actually representing the codon. Now, this is the first thing. The second thing is the ribosome. This ribosome, now the ribosome, this is the 30S, fine? 30S. And after that, 50S subunit will bind here. Inside the ribosome, there are the 3 sites. This one is called A site, we can say amino acyl site. This one is called P site, we can say peptide site. And this site is called E site, we can say exit site. Now, first of all, what actually happened? No, this is the second thing. This is the second thing. The third thing which are involved in a protein synthesis, this is actually the transfer RNA. So let me draw here what actually how it will be look like. It will be look like, and they carry the amino acid. Now this transfer RNA contain anticodon. We call that anticodon. Anticodon. And this black one, as I draw on the messenger RNA, we call that codon. So when these three things are bound together, or when these three things are combined together, we call that a initiation complex. Initiation complex. So this amino acid, this transfer RNA will bind to the P site, not with the A site. So what are actually happen when all these three things are combined together? Then we call that, collectively we call that initiation complex. How this initiation complex is actually bind? First of all, from DNA, or we can say from DNA, or transcription is occur due to which we get a we get the messenger RNA. This is messenger RNA. We get messenger RNA from transcription. Now what actually happened, as I told you, this messenger RNA contain codon. First of all, there is the 30th subunit will bind to that area. And this is the 30th subunit. Now what actually happened? In 30th subunit, this is the A side, this is the P side, and this is the E side. With A side, there is a release factor bind somewhere. There is a release factor. So this is called release factor. RF means release factor. So at somewhere there is a release factor bind. What are the main function of this release factor? The main function of this release factor that they prevent, or they not allow the 50th subunit to bind here. First of all, what actually happen, the transfer RNA which carry the amino acid. So let me draw again. And now this is the transfer RNA which carry the amino acid. Fine. This first amino acid is actually called F-met. Or we can say formylmethionine, or we can say that this is the first transfer RNA which carry first amino acid, and we call that F-methionine. One thing you should have to keep in mind, this F-methionine, or we can say this, now when the amino acid contain the, when the transfer RNA contain the amino acid, we call that charged transfer RNA. So this is actually charged transfer RNA. This is actually charged transfer RNA. When they lose the amino acid, then we call that uncharged transfer RNA. What actually happened? First of all, this charged transfer RNA will come and bind here. So this is the codon area, and with codon there is anticodon of the transfer RNA. So this is the transfer RNA, and then they carry their amino acid. We call that F-met. Now after that, when they bind to the P site, then what actually happened? This release factor will release out. Let me draw with a red marker because it is already in a red. So this release factor will release out. Once this release factor will release out, then what actually happened? Then 50th subunit are bind to that area. So this is the 50th subunit. So 50th subunit are bind to that area. After that, when the 50th subunit are bind to that area, then again 50th subunit will carry three subunit also, three site also, P, A, and E site. Now what actually happened after that? After that, another new charge transfer RNA. Let me once again draw here. Then another new charge tRNA will bind to this A site. So new charge tRNA will bind to this to that area which carry the amino acid. Now when both sides are filled by the transfer RNA, then this ribosome will move forward because the ribosome have the movement ability. This ribosome will move forward. This ribosome will move in this direction. Once the ribosome will move in this direction, we usually call that translocation. We call that translocation. Translocation. So we call that translocation. Then what actually happened in translocation? In translocation, this amino acid which is present with the transfer RNA on a P site will cleaved. And they will shift or attach with this one. They will cleave and attach with this amino acid of this new transfer RNA. And the transfer RNA which is present on that side, we call that uncharged transfer RNA. Why? Because they lose their amino acid, or they denote or they shift their amino acid with the A side transfer RNA amino acid. Now after that, what actually happened? This uncharged transfer RNA will migrate to this E site. So this is E site, and then they will expel out, having no amino acid. So this is the uncharged transfer RNA. Uncharged tRNA. So this is uncharged tRNA. They will expel out. Now once again, what actually happened? The transfer RNA which is present on A site during translocation, the transfer RNA which is present on A site will shift on the P side, and the transfer RNA which is present on P side, we usually call that uncharged transfer RNA. They will shift to E side and then expel out. So how it actually will be look like? So the whole diagram will be look like in this form. This is the 50s. This is the 30s. These are the site. These are also the site. So this one transfer RNA will shift to that area, having the amino acid of the previous one and having its own amino acid as well. So these are the two amino acids, and this uncharged transfer RNA will shift to this E site, having no amino acid, and then finally they will expel out. And again this A site, again this A site is free, and then again another new amino acid, we can say new charged tRNA will come and they will bind to that area. Then again the amino acid which are present with this transfer RNA will shift to with new transfer RNA, and again we can say again the whole process will repeat it once again. Fine. So once you get from the beginning, then the rest of the transfer RNA which are coming will be very easily for your understanding. So this is the main process of the transfer RNA. So when the transfer RNA are coming and when the amino acid are binding, then what actually happened? Finally, there is a stop codon are coming. This process will be continued until a stop codon are not come. Once the stop codon are come, let's suppose this black one represents that these are the stop codon. Then what actually these are? Let's suppose these are the stop codon. Then what actually happened? In stop codon, let me draw here with a blue marker the ribosome. In stop codon, as this is the P site, let's suppose, and this P site will carry the transfer RNA having the very long polypeptide chain, or we can say short, or we can say, now these are the polypeptide chain. We can say polypeptide chain. Finally, a polypeptide chain is formed because a lot of amino acids are binding and they make a very long chain. We call that polypeptide chain. Now once this polypeptide chain are formed, and when the ribosome reach to the stop codon, then what actually happen, this will cleaved away from the transfer RNA, and this transfer RNA again expel out from the exit, or we can say E site, and then this polypeptide chain will code for the protein, or we can say this polypeptide chain will make their structure and make a protein which are used in a bacterial cell for different purposes. So this polypeptide chain is actually, we can also say that this is actually the protein. And it's very complex and very large topic, but I explain it very simply, fine. So this is the protein synthesis. So that's all about the protein synthesis, that how a protein is synthesized inside the bacterial cell. Once you get the idea about the protein synthesis, that how actually it is synthesized, then inshallah in the next video the mechanism of action of these drugs will be very easy for you. Fine. Inshallah in the next video we are going to talk about the mechanism of the action of each of the drugs, the resistance, mode of action, and antibacterial spectrum, and also the side effect of these drugs in a detail. Fine. So for this purpose, please subscribe the channel, and if you like this video, please like the video, and if you have some problems, or if you have some question related with this topic, so write it into a comment. I will try my best to give you answer as soon as possible. So please like the video, share the video, and also subscribe the channel to get more interesting video lecture like that. Thank you so much.